Hello, welcome back to another science lesson. Hopefully, my mic works. It does work. Okay, so today we are going to discuss some science techniques. So, first, you can see how it says there Western blot, but let's go with immunoprecipitation first because that's what I have on this page. So, where's my marker? Does this work? Yes, it does. So, no, eraser. Make the eraser that one. Okay, so we have immunoprecipitation, right? Immunoprecipitation. If someone asks you, what is that for? It is used to isolate. Like, we use it to isolate a specific protein, okay? So, immunoprecipitation isolate specific proteins and its partners and its interacting partners there from a complex mixture using an antibody. So how does that work? Essentially, the antibody binds to the target protein, and then the complex is pulled down using beads. But it's easier if I just draw it out. So let's say you have an Eppendorf tube, right? This is the tube. It's closed right now. And you have a couple of proteins in there. You have some red stuff, and then you have the blue stuff. No, let's make it purple. The drawing is purple. No, that's gonna be hard to see. Let's make blue. Here. Blue stuff. There, perfect. So those are proteins. But the blue one is your... No. The red one is your protein of interest. No. Here. Let's say the red one is your protein of interest. Protein of interest. And then, so you have your cell lysate right now. Cell lysates, that's all of your proteins. All proteins or total proteins. Then you put antibodies. Let's make it green. But my drawing says blue. Okay, antibodies, they look like Y. Like that. Those, you add the antibodies and you incubate them. And then, in your Eppendorf tube, what happens is, if you zoom in, let's just zoom in. If you zoom in, we had this, right? These pictures. Your antibody would now attach to the red one. Hello, Brazil. Welcome to the stream. Oh, yeah. If you type in the chat, you get a mini character and you can actually change your hat. Ohayo gozaimasu. Hat. So if you type like this, exclamation hat, you can change it. I'll wait until mine pops up. See, right now my hat is, what's that? Like a signal tower. I want to change it, so I'm going to type exclamation hat, and boom, it'll be a different hat. Okay, so I said right now we have your total cell lysates, the all proteins. You put your antibody in it. So now you have, and then you incubate it. So now you get your antibody, antigen, antibody complex. Okay, aka just the things if you look at this one, your antibody is now connected to your antigen, the one it wants. After that, you put beads. You put the beads, protein beads, and incubate. Ooh, you got the haha hat. Perfect. I want mine to be the dab or sparkly, so I'm gonna do that too. Okay, after you incubate with the beads, your beads will now attach to the antibody. So that's your test, that's the Eppendorf tube. And then if you zoom in again, here, this is the red one's the protein that you like. I said after you put your antibody and then it attaches here, and then with the beads will attach to the antibody here. So now you have beads attaching there, and then you wash. Is this doctor stuff? And anyway, yes, right now it is experimental techniques, experiments. We are trying to isolate a specific protein. So this is the protein. These are the proteins inside the Eppendorf tube, and then we're just trying to get the red one. So how do we do that? Put an antibody, and after that you put in the beads, the protein beads, and then after you wash and elute, you will yes, thank you very much for coming. Yeah, if you elute just means you let things, you wash it off like that, make it all flow. What the remaining thing inside your Eppendorf tube after washing is, guess what? It is only your protein of interest, the beads. That is your protein of interest, your antibody, and then your beads. That's the ones that are left. And then you just purify it, and then Western blot, using the Western blot is the one that looks like a tower with these bands. So that is immunoprecipitation. Okay, and then if you want to do co-immunoprecipitation, it's similar, but take note, immunoprecipitation, just the word immuno, it's just isolating the protein that you want. But if it is co-immunoprecipitation, precipitation, then that would mean, what's that called? That is isolating a protein with its binding partners, aka it is protein to protein. Isolate the protein, and funny enough, it has another protein attached to it. So it is same as immunoprecipitation, but you have a bait protein in a complex with other proteins, and both proteins would co-precipitate. So how does that work? Again, we have our Eppendorf tube. We try to zoom in. If you zoom in, this time I will try to draw a couple more proteins. This is the protein that we want. I'm making it red just to make it as obvious as possible. We have the blue one there, and then with the red one, you have a mini purple one. A mini green one attached to it. I used green earlier, so I'll just use purple. There, you have a purple one attached to it. So if we're talking about co-immunoprecipitation, first you add antibodies to your cell lysate, aka the total protein. Again, the antibody looks like a Y. Now the picture will look like this. You have the proteins that you're not interested in, and then the protein that you want with something else attached to it. Guess what? The antibody will bind to this. There you go, attaching to the red. That's your primary antibody. After that, you add the beads. After you add the beads, the beads will now be in there. Again, how does it look like? You have your protein of interest, you have the extra one, and then you have the other protein that attaches to your protein of interest. You have the primary antibody attaching to your the protein complex of interest, and then you got the beads. Hello, thank you for coming by, WJ21. Oh, you got the wave emote. I will try to change my hat also. You can change your hat using exclamation hat, because mine is a pizza hat. I want the dab hat. Dabu. Okay, now what is this? We added the beads, and then we elute it. How can we elute it? Yeah, you just wash it off and elute. And then now what's left here, oh, I forgot to draw the tube. And then let's draw the tube again here. Circle. Okay, now if we wash it off, we're removing the stuff that the primary antibodies did not bind to. So no more blue ones, only the purple and red ones with both the beads and what's this called? The primary antibody. Oh, I should type a mini note. How can I add a mini note? Timer has some special features, aka that one. That's a mini. I will pin that one here. Pin message. Okay, so if you wash it off, you remove the other proteins that you don't need, and then after that you will just purify it. You wash it, and then after you purify, the impurities are gone, and then you just use Western blot to check it out. Alright, so now we did the co-immunoprecipitation and protein. Okay, now the way that works is, let's see, we finished that one. Wait, yeah, so far we did immunoprecipitation, aka just immunoprecipitation, and then there is co-immunoprecipitation. And that one is just the protein one. And then guess what? There is RNA immunoprecipitation, and that one is called RIP. It's called RIP, and that is to identify the RNAs bound to a specific RNA binding protein. So it's RNA protein complex. Got 'em? Okay, so if the main mechanism for this is an antibody against the RNA binding complex is used to pull down the RNA protein complex, and you will just discover the RNA targets of the RNA binding protein. So how does that work? First you have your cell, and then you cross-link the proteins and you cross-link the proteins and the RNA and proteins cross-linked. After that one, you lyse it and fragment, and then you immunoprecipitate by like putting the AB and putting the beads, and then you purify, and then boom, what's left is you have your protein, and you have your RNA, and then you have your protein. So that's the main difference of it. RNA protein is just you have it, the thing it's attached to is the RNA. Then now you have chromatin immunoprecipitation. That is just protein and DNA, and similar steps kind of with RNA immunoprecipitation. That is, you have your cell lysate or cell lysis. Let's erase this one. You have your cell to study it. You use it to study protein DNA interactions in vivo. And then the mechanism is you cross-link proteins to DNA first. This is chip. Chromatin. What is that emote? Is that salt? I'm not sure. It's okay. Chromatin immunoprecipitation. Precipitation. Wait, is there this one? Yeah, that is chip. Chromatin immunoprecipitation. Oh, copying. Oh yeah, that is funny. This all sounds like witch talk. Prepare the lake. Welcome back to the stream, Yun Yang. Yeah, we are trying to isolate DNA and protein now. Right now we are in chromatin immunoprecipitation or chip. So, and this is in vivo. First, we cross-link the proteins to the DNA. So this is your DNA, and then we have proteins. We cross-link it. Now it is linked. That could be inside a mini plate, but after a while, you have this. You have your antibodies, AB, and cross-linked DNA proteins, because you will add, what's that called? You will add, what was it? You add your antibodies to it and then incubate after. Nice, that one. After you incubate, you just continue doing that one until you elute it, and then you get your cross-linked, right? Isolated, the proteins and the DNA, and then you just fragment it by reversing it. And then now you have it. You have the solo one. You just purify it after. Now, there is something called fast chromatin immunoprecipitation, and that's just same as chip, but you optimize it for speed. So you first have your cell lysis and your total DNA purification, and you have your antibody plus DNA incubation and binding. And then you wash it and reverse the cross-link, and then boom, you have DNA lysis. Haha. Okay. Now, we talked about RNA immunoprecipitation, right? But what about RIP-seq, RNA immunoprecipitation, sequencing, or cross-linking and immunoprecipitation sequencing? So let's draw this, how it works. Let's see. What is this goal button? Wait. Let's see. Does that work? Oh, okay. I don't know. It could be. We'll try that later. But for now, let's focus on RNA immunoprecipitation sequencing. Okay. So first, let's see. Number 1, you do in vivo cross-linking. In vivo UV cross-linking. Cross-linking. And this is where your proteins are irreversibly cross-linked to RNA. Because take note, we are doing RNA immunoprecipitation sequencing. RIP-seq. Okay, cross-link it first. What does that mean? I keep on saying the word cross-link. It sounds funny. So right now we have your RNA, and then you have your RNA binding protein. If you cross-link, that means UV. You can use UV to attach to induce the cross-linking. Now you have to do cell lysis, aka the cells are lysed and the RNA is purified. So that would guarantee that you you only have your RNA binding protein of interest. Here. Take note. Let's actually write an extra one. This will help us out later. So you have your orange and you have your red. The red one is something that you don't want, but for now, let's say it's also cross-linked because it won't do cross-linking. So now, if you zoom in your cell lysate, you may have some RNA like this. That's your RNA. And then you have the orange. And then your red. Let's see. Red and red. After that, after you lyse and RNA is purified, you will do partial RNase digestion. So the only ones left will be the RNA that's attached to a protein, whether it is the RBP that you want or not. But the rest is, you just get rid of it. After this, you purify, aka how do we do that? You do immunoprecipitation. How? We talked about this before. What we'll do is the target RBPs are purified with an antibody that you want. So let's say the yellow one, which one is it? Yeah, let's say the orange one is the one we want. What will happen is, if we put in the antibody, let's say the green one, if we now zoom in here, you have your RNA, and then you have the orange one that we like. Oops. The red one that we don't like. And then you have your antibody attaching here. So later on, when we wash it off, only this one is left. And then now we have 3' adapter ligation. This is new. 3' adapter ligation. So the adapter is ligated to the 3' end. What will that look like? So you have 5', 3' end of your protein, your RNA. You have your protein over here, the RNA binding protein. You have your antibody here. And then you have an adapter. I will just make it red. You ligate it over here. And then what's next? What's next is you do SDS page. SDS page is just gel electrophoresis, but you wrap it all in a negative charge, so only the sizes matter for that one and not the charge. But how it looks like would be like this. This is the tower. You have IP, and then you have RBP and control. For control, I have to move. Okay, I will draw this out properly because it will help us analyze the bands. Okay, let's draw it this way. So you have your SDS page here, right? I'm just estimating how many wells I need. So you did immunoprecipitation. We have control over here, and you have RNase. So if control does not have RNase, and here for, oh, this is the ladder, normal ladder, to check the size. For control, there's nothing here. For RBP, if you have plus plus and you have just a tiny bit, for the plus plus you have it, the band here, and then for the normal plus you have the band just a little smudgy here because you only added a tiny bit. And then this band would mean that is the RBP bound band. This is the band where you have your RNA binding protein. Because it took some time for it to like go. I mean, well, it traveled faster, but yeah. Now let's do proteinase K digestion. Proteinase K digestion. So the RBP is digested and you only have your cross-linked polypeptide, aka from a very big protein, the very big RBP, now it's only the exact polypeptide attached to your RNA. This is the one you want. Now you have your protein, right? And then you have this RNA. We can now do reverse transcription to make that back into DNA or cDNA, complementary DNA. And then you do PCR to do Tajukagi Bunshinjutsu times 2 times 2 times 2. And then just do sequencing. So that was a lot, but essentially that is RNA immunoprecipitation sequencing, where first you cross-link. Well, the goal of RNA immunoprecipitation sequencing is you have your RNA and protein, and then you want to find the specific RNA your protein is interacting with. So you the cross-link is induced with the UV, and then you put in antibodies, which will drag. After you drag it down, you now have your RNA bounded with a protein, and then you will do RNase digestion, just partial, so that only the specific ones that are attached to the RBP is the one that you get. And then you add an adapter, and then you do SDS page, and now you have your specific band that you want. And then after that one, you just convert the RNA into the cDNA. Next, we discussed so far immunoprecipitation, and now let's do flow cytometry. Flow cytometry. Flow. Okay, so for flow cytometry. The way flow cytometry works is that you analyze the physical and chemical characteristics of the cell. Okay? So for this flow cytometry, I will write it. You analyze the physical and chemical characteristics of cells. So that in here your cells are labeled with fluorescent antibodies or dyes, and then let's say you have your cells like this, and then you pass them, you have a lot of cells, right? But then you pass them through a laser. This is the tube which goes just one cell at a time, and then you have a laser which hits it, and then you have a sensor which will analyze its forward and side scatter, and that's when you know like the height or length of a protein. But with that you can actually check or quantify how many cells you have exactly. So that's it's mostly if you want to do if you need to do quantification, that is where flow cytometry is very useful. Next is the type, not exactly the types, just specific versions. So we mentioned flow cytometry, but there's also something called fluorescence activated cell sorting, and magnetic activated cell sorting. So we're moving to the CS, cell sorting. Earlier we mentioned flow cytometry, and then a specific flow cytometry version is fluorescence activated cell sorting. But magnetic activated cell sorting is not a type of flow cytometry. Okay, so just make sure you don't mix that up. So for fluorescence activated cell sorting, you sort the cells based on their fluorescence. So here, earlier it looks like this, where that you have a lot of cells and the cells go like this. But in here, before you make them go there, you put a fluorescent antibody. So there are some cells that have your antibody, this cell and then this cell, and then they just get sorted into different tubes. Let's say this is plus tube and this is minus tube, negative plate. So your cells that are more positive will obviously go to the negative plate, because like opposites attract. So that is how it works. Population of interest versus your other cells that you don't need. But the thing is, if you don't need to do cell sorting, then just do normal flow cytometry. But what is magnetic activated cell sorting? This one, you isolate the cells using magnetic beads coated with antibodies. So let's say you have your cells in a tube. You label the cells of interest with magnetic particles. And then after that one, you prepare your magnets. Oh, this is too tiny. Let's draw it here. So you have your cells right here, and then you have magnets. Let's say it's like this. The magnets. Magnet 1, magnet 2. The way you use that is you apply labeled cell suspension to the column and collect the flow through. Okay, so you throw it in there, and then slowly but surely you collect the ones that you don't like first, because the ones that you like are sticking here to the magnet section. So this is the first ones that are flowing through, are not the ones that you like. After that, you wash it with a buffer just to make sure only the ones you like are left, and then you just remove the magnets and then collect it. So that is how magnetic activated cell sorting works. You isolate the specific cells using beads with the antibodies. Yeah. So now, I'll drink water first. Alright. Oh, I'll increase the thickness of my marker to make it look, like to make it easier to see. Yeah, this one works. Next is pulse chase. Pulse chase, pulse chase, pulse chase. Here. Pulse labeling is the first one. And then you have pulse chase here. So for pulse labeling, that one is you label newly synthesized molecules over in a short time window. Label the molecules. Label newly synthesized molecules. Molecules. So how? Well, you add a labeled precursor. So for example, this is your cell. You have a lot of amino acids like this. Because this is, imagine you're making a protein, right? This is your ribosome. This is the protein that's being made. Here, squiggle. So now you pulse with radio labeled amino acids. And then now the one here, erase first. The one here is your radio labeled amino acids. Here. So I'll make it red so it's easier to see. Now you switch to an unlabeled precursor. So after that one you chase. This will follow the fate of a labeled molecule over time. Here. So you have your radio labeled protein, slowly but surely. But then after that, if you put the non-fluorescent amino acids, then the next ones it'll make are not red anymore. So that one will help us check the protein turnover, and that's why there's like a time element for it. You isolate the protein and measure the radioactivity. So if you want to check, like both use like fluorescence, but pulse/pulse chase is not a time course experiment. I mean, pulse and pulse, okay, I was talking about Western blot versus pulse chase, okay? So both are both use fluorescence, but pulse chase has a time element. So you can actually combine both, where you label proteins by pulse, and then after that you run a Western blot to detect the protein of interest using time and specificity. Okay, because if you remember, Western blot is, did I talk about it? No, not yet actually. Okay, so proteins, if you do SDS page, you separate them by size using SDS page and then after that you transfer to a membrane. The membrane will, and then after the membrane you put the antibody. Now the membrane will just help it stick, like you should fix it, and after fixing you put an antibody to the specific protein that you want, and then the secondary antibody will bind to the primary antibody, and then after that you put you detect the fluorescence. So both are fluorescence, but the secondary antibody is just for detection and not exactly to track it. So it's just a snapshot of how much of a protein is there during a specific time, not exactly during all time slots. Okay, and then next is gel electrophoresis. I think I mentioned this multiple times. I'm just gonna summarize it a bit, because there's 4 terms right now that's floating. Here. So far I mentioned gel electrophoresis, And then there is also a thing called SDS page, which is a type of gel electrophoresis, the GE. And then you have 2% agarose gel electrophoresis. Take note, these are types of gel electrophoresis. The purpose of that one is just to separate charged molecules using an electric field. So it's usable for DNA, RNA, and protein. SDS page is sodium dodecyl sulfate polyacrylamide gel electrophoresis. This will separate proteins by size, aka molecular weight. So you denature the protein and coat with, so denature the protein and then you coat it with an SDS. So if this is your protein, yeah, that's your protein. You coat it with SDS, which will help it get like a lot of negative charges. You coat it. So now only the size of the protein will matter and not the charge. It's just very coated with negative. And then you now pass it through the gel. Oops, wrong color. Pass it through the gel. This is the gel. You have a buffer. Imagine it's lying down, but you have a negative charge and positive charge here, and then it will just flow. The smallest, if I remember it correctly, I think the smaller fragments, yeah, will flow faster. So meanwhile, the bigger proteins will be very slow, like that. So the bigger ones are above here, and the super small ones are there at the bottom. That's SDS page. Meanwhile, the 2% agarose gel electrophoresis, that does not have your SDS. So you dissolve the agarose powder in the buffer, maybe TBE, so buffer, and then you make your agarose gel. You load your samples in here in the wells, and then you apply the current to pull the DNA toward the positive electrode. So negative is cat, positive anode. And then why will DNA go towards the anode? It's because DNA is naturally a little bit negative, okay? Why? It's because of the phosphate bond. If the phosphate is over there. Why is phosphate there? It's because there's nucleotides at the bases here. Because if you zoom in, I think I have a mini drawing later on, but you have your DNA structure like this, and then the things in the middle are the base pairs. Yeah, we drew it in the old one. And then if you just zoom in, you have the different termini. Yeah, you have different terminuses, 5' and 3', 5' end and 3' end. Yes. Okay. So far. Oh, take note. This is also where nucleotide and nucleoside comes in, which I talked about in my previous one. It's like you have your sugar, and then your base, and then your phosphate. The phosphate is where your negative charges are, and then nucleoside only has the sugar and the base. Okay. Let me just write this really quickly. DNA and ATP structure. Nice. Okay, so now we have Western blot, detecting the presence and amount of a specific protein. Erase, let's erase it. So for Western blot, this one here, Western blot, this one is detecting the presence and amount of a specific protein in a sample. So you do SDS page, transfer to a membrane, add the primary antibody, detect using fluorescence, and boom, you have it. So Western blot's purpose is just to check out your proteins. And then we have the 4 types of blots. Hello, Antwerpen. Thank you for coming to the stream. Please type in English if possible. I cannot understand that. And the next is the types of blots. Here. Oh, by the way, you can change your hat using exclamation hat. And I will actually try that. Hat. Like that. Okay. So for the types of blots, let's write them all first. You have Northern blot, Western blot, Southern blot, and then Eastern blot. Some of these are more common compared to the others. So I'm just gonna write the arrows for now. What does that mean? This means Southern blot is to detect your DNA sequence or fragments. So this one, hello Apu, yes, hat. For Southern blot, this one is DNA would be isolated from the cell. You cleavage it. Now it is small fragments. Then you break them down and isolate it. You separate, denature, transfer, hybridize, and visualize. Essentially, the main focus is just Western blot because it's more common. Western and Southern. Extract the sample again. Yeah. After that one, there is the Northern blot, which is for RNA. Extract, and then denature, separate. So essentially it's extract, denature, separate, visualize. Something like that. And then the Western blot is for proteins. It's also known as immunoblotting. We talked about this. How does a Western blot work? First, you have your cell membrane, your cell collection, and then you lyse them. You lyse it, and then after you lyse, you do the SDS page to separate it based on just their molecular weight. And then after that one, you visualize the proteins using dyes. Don't forget the dyes. After that, you blot and transfer to a membrane. After you separate it, put it on the membrane. After you see the membrane, you visualize it using the antibodies. Okay, the primary and secondary. And then you see it. Meanwhile, Eastern blot, not super common, but it is for post-translational modifications. And that is where you see like glycosylation or phosphorylation. Yes. Okay? That's what you see there. Okay. So now our next thing on the agenda is to do a mini review. So we have a couple of questions here and we'll go through all of them. Well, some of them. So let's see. Erase. So for here, what does this one mean? Alright, here, let's start with what's the difference between a selection versus a screen. So a selection only allows, selection allows only the cells with a desired trait to survive, while the screen allows the cells to all cells to survive, but it will require the researchers to identify the desired ones manually. The selection is harsher. Only cells with the desired trait will survive. So if it is in Naruto form, only the ones who can do Sage mode will survive. The rest are killed. Okay, next, if it is a screen, all the cells will survive. Let's say all the red stuff and all the green stuff will survive, but and then it will require the researchers to like pick out the specific ones that it wants there. Meanwhile, in the selection, you only these will survive. Okay. Next. Oh, actually, we can give an example for that. As an example would be lactose metabolism, wherein only bacteria which can use lactose will survive on lactose-only media. And in a blue-white screening on X-gal plates, all colonies grow, but blue colonies express LAC-Z and whites do not, and the researchers would pick out the white ones. Hello, thank you for coming back, Elmer Alejandra. Yes, we are here today discussing research methods. And so the next question is, what does it mean to say if two genes are orthologous? Orthologous. And what's the difference between percent identity and percent similarity? I wonder what that means. So for orthologous, that would mean the genes in orthologous genes are genes in different species that evolved from a common ancestral gene by speciation. That was a lot. So it just means if you have this gene and then by speciation it splits up into like two different types, then you can say these genes are orthologous. Meanwhile, percent identity, it's the exact match of amino acids. Percent identity is the exact match of amino acids. Orthologous genes are genes that came from like an ancestral gene and just split up by like speciation. Meanwhile, percent similarity includes conservative substitutions. Like for example, you have similar amino acids like the leucine and isoleucine. So leucine looks like, I think if I remember correctly, straight N plus and then COO minus. We have valine before leucine, gallium. So this one is CH2. I think like this, and then it splits up. CH. Yeah, because valine is just like this, immediately splitting up into methyl. Yeah. Isoleucine looks similar to leucine, but at the end it has C, so an L, a straight leucine. So again, percent identity refers to the exact match of amino acids, while percent similarity includes the conservative substitutions of it. Yet, so how are tissue or cell type specific, transgenic, and knockout mice generated? Where's my marker? There it is. I wonder how. Let's see. There, ready. So the question was, how are tissue or cell type specific, transgenic, and knockout KO mice generated? So tissue specific mice are generated if you insert a gene of interest downstream of a specific tissue promoter. What? Okay, tissue specific transgenic mice, aka the mice. This is a mouse. No, look at that. Whiskers. This is a mouse. It looks like a cat. Oh no. Oh no, it looks like a cat. Now it's just a cat. No, I want what does a mouse look like? Kinda like this. Now that's just a bird. Okay, let's just say you have a mouse. That's a mouse. So that mouse, the tissue transgenic mouse, it's generated by inserting a gene of interest. You have a gene of interest downstream your promoter somewhere here. So that is how it is used, and the gene is flanked by LOXP site, and it is expressed under a specific tissue promoter, and excising the gene only in that tissue. Okay. Now, what is a PCR? Polymerase chain reaction, its major uses, and what does real-time PCR measure that standard PCR cannot? So we talked about this before. PCR versus real-time PCR. So PCR would just amplify the DNA sequences through the cycles of denaturation. So you have denaturation, primer annealing, and extension. So it's used for cloning. Meanwhile, RT-PCR is real-time PCR, or qPCR, measures the DNA amplification during the reaction. So it is quantitative. Meanwhile, normal PCR would just be to show presence or absence of it. So again, PCR versus real-time PCR. PCR would just show the presence or absence. Meanwhile, real-time PCR would quantify its the amplification. Next is explain biological replication. And technical replication. So biological replication, you will use biological versus technical. Biological replication will use independent samples to check the natural variability. Meanwhile, technical replication repeats the measurements on it to assess the precision. So again, biological versus technical. Biological will just show, or will use the biological samples. Okay, what is meant by genotyping? Genotyping will identify which genes an individual carries at a specific gene locus, often using PCR sequencing or hybridization. Again, genotyping will just identify which alleles an individual carries at a specific gene locus, mostly using PCR sequencing or hybridization. Why should RNA be rapidly stabilized for transcriptomic studies? And why is it a bigger problem for RNA compared to DNA? Well, especially in bacteria. That's because RNA is unstable. And that's why, because it has a 2' hydroxyl group, making it very prone to hydrolysis. So if we want to double check the structure of an RNA versus DNA, here. RNA versus DNA, specifically, it's sugar. That's because you have DNA deoxyribose sugar, while RNA has ribose as a sugar. So if you write their sugar molecules, both starts with a ring on oxygen over there. Deoxyribose, you have up, nothing, down, and this is methyl, and then OH. That's where the proteins are. But in RNA, you have OH, and you have another OH down here. Double whammy. So this is the 2' OH. It means it's very reactive over there. And that's why you need to stabilize it first. Bacterial cells have higher, what's that called, reactivity. RNA has higher RNase activity and have faster RNA turnover rate, so that's why. Stabilize them, it's a very urgent step that you need to do. And what is a restriction modification system, and how is it used in biomedical research? A restriction, let's erase it. Here. There you go. So a restriction modification system, if we're being funny, that one is a restriction modification system protects bacteria by cutting foreign DNA with restriction enzymes, while methylating their own DNA for protection. What does that mean? Well, it will cut. So in research, restriction enzymes are crucial tools for cloning and DNA mapping, right? So there, you cut the foreign DNA, cut it, while methylating your own DNA. You cut and methylate. What is CRISPR-Cas system? We talked about this. CRISPR-Cas is a system where you use, what is CRISPR? Clustered, regularly interspaced, short palindromic repeats. So CRISPR will use some guide RNAs, yes, to guide the Cas9 to cut the specific DNA sequences. Hello Kaizaki, welcome back. We were just talking about CRISPR again, the clustered regularly interspaced short palindromic repeats. Ayo Landor, welcome back. Hat, yes. I think you might have to wait until your character pops up actually before you can change the hat. There you go. Yeah, that's the hat. But yes, that is CRISPR. Clustered regularly interspaced short palindromic repeats. Yeah, you have to wait until you're wearing a hat before you can change your hat. Clustered regularly interspaced short palindromic repeats, and in research it will enable precise genome editing so that you can do knockout, insert, or modify genes in various organisms. Now, what is the basic structure of an IgG antibody molecule? I wonder. I still haven't done the mini lecture for the IgGs, but I will draw it now. IgG structure. It looks like a Y. Okay, I was about to write, unless he can change things that didn't appear yet. Oh my gosh, that's true. Like before the character even jumps in, it gets confused. Like, I have to change the hat now. I'm not wearing anything. I'm not wearing a hat yet. Yeah, so that's how you confuse the enemy. IGG. It looks like a Y, like this. Double Y. Beautiful. Look at that. Oh, actually, we can try and make it even more exact. Yeah, since we're crazy, let's just keep it up. So we have the blue ones, right? Over here. You have the blue one over here, and then the red one, or the pink one. There's a reason why I'm using the light colors, trust me. This is blue and pink. And then you have the darker blue over here. Dark blue. And the red one over here. Why? I'll explain it in a bit. Let's write the yellow also over here. Over here. And then an orange one. A dark yellow here. Lastly, light green. And then dark green. Okay, so what am I writing? This is the IgG structure. You have the variable region, and you have the constant region. The big ones are called heavy chains. These are heavy chains. And this is the light chain. And then you have the antigen binding site, which is this antigen binding site. Yahat. So IgG antibodies have 2 heavy chains and 2 light chains. Linked by what? Linked by disulfide bonds over here in the middle. Disulfide bonds. The variable regions at the tips of the arms form the antigen binding sites and will determine the antigen specificity. That's why it's called variable region, because it can change. Okay? And what are the key differences between primary cell cultures and cell lines? Primary cultures are derived directly from tissues. Okay? Wait. Yeah, there you go. Primary cell cultures. Let's erase it. Oh, it reminds me of that group, the Erase. Their cover of The Ice was great. Erase this one. Oh, no, my screen is just dirty. Okay, next. What are the differences between primary cell cultures and cell lines? So primary cell cultures are derived directly from tissues. This is from tissues and closely mimic in vivo behavior, but the time span is limited. Low HP. Meanwhile, cell lines are immortalized and divide indefinitely. However, it could diverge biologically from normal cells, so that's why sometimes primary cell cultures are much better compared to cell lines. But for longevity, cell lines last longer. Next is, how does Sanger sequencing work? And what is a dideoxynucleotide? Okay, good question. Wait, before that, let me drink water. Water. Okay, I don't want to know. How does Sanger sequencing work? The way Sanger sequencing works. Oh wait, I forgot to mention. Funny thing is that timer could increase with with like subscribers, supas and members. I just yeah, why did I just mention that now? But that's how it works, but it's okay. I'll just continue reading science as well, even though it'll reach there if ever. But yeah, how does Sanger sequencing work and what is a dideoxynucleotide? Sanger sequencing uses DNA polymerase with normal and dideoxy DDNTP, dideoxynucleotides DNTPs. That one lacks a 3' hydroxyl group, which terminates DNA extension. Okay? And if you analyze the terminated fragments, the DNA sequence is determined. Booyah! Let's go. So that's how it works. What is next-gen sequencing and how does it work? Like Illumina. So in Illumina sequencing, I think, yeah, in the next mini lecture, I will talk more about this one. This one's just a quick introduction for it. In Illumina sequencing, millions of DNA fragments are attached to a surface, and that's amplified into clusters and sequenced simultaneously by incorporating fluorescently labeled nucleotides one base at a time. Okay, that's how it works. In a protein purification, what is an affinity tag? Affinity tag is just a small peptide or protein added to the protein of interest to aid the purification. Again, so what is an affinity tag? Just a small protein, like an ID, but it's in the form of another protein, which will just aid the purification. So an example is the histone tag will bind to nickel columns, and it is eluted with imidazole. Again, the histone tags will bind to nickel columns, and it is eluted with like imidazole. And GST tag binds to the glutathione resin and is eluted with glutathione. Now, oh no! Oh, there it is. Draw the next one. It is about the last lecture. Draw the structure of any of the 20 standard amino acids, and then of a dipeptide to show a peptide bond. Let's see. Do I remember all 20? Oh. If I don't, I'm gonna have to grab my notes or watch the previous lecture. Ha ha. Har har. Okay, let's see. Structure 20 amino acids. Oh no. No, it's okay. It's fine. It's okay. 20 amino acids. First, we have the non-polar amino acids. Glycine, Gly-G. This is just HCCOO minus NH3 plus H. Alanine, ala A. Just it's N0 minus H3N H CH3. Valine, Val V, CH H3N plus COO minus CCC. Next is leucine. I'll write it here. Leucine, L HCCOO minus H3N plus. Leucine is just a longer valine. Isoleucine is ILE, and then that is CHCOO minus H3N plus CCCC. Okay, next is I need space. Gav, Li. So we did Ga, Gav, Limp, FW. Methionine is the next one. Methionine, Met, M, HCCOO minus, H3N plus, methionine, CCSC. Yeah, I think. Is it? One moment. Check. Just to make sure. Methionine structure. CCSC. Good. Very nice. Okay, next is proline. Oh, P, the ring. That is literally the ring. This one is CHCOO minus, H2N plus 1, 2, 3, N. Beautiful. Then we have phenylalanine, F. That is, wait, COO minus, H2, 3, N plus. This one is C, H3, and a benzene ring. That's phenylalanine. And then you have tryptophan. Right? It's tryptophan W, right? Tryptophan. Tryptophan. That one looks like this. C, HCOO minus, H3N plus, C something. You draw a house, you draw a hexagon, and H. This is H2, that means it's H2. Bond. I know there's a bond. Where is the bond? Oh, this one is obviously a ring, right? Okay, I think it's this one. 1, 2, 3. No. Just to double check where the bonds are for tryptophan. Ring, boomerang. Oh, it's okay actually. We don't need to write the last bond because this is already taking it into account. Okay, so we did got 9 out of 20. What was the next one? We have polar. Uncharged, and charged. Uncharged is stun QNY. Stun QYC. Okay, what are those? Serine S. This one is. One moment. I'll erase so I have space. That rhymes. Serine space. HCCOO minus, H3N plus, serine C. OH or SH? SH? Okay, now I know I have to check this one. Serine structure. Oh, it was OH. COH. The SH is the last one later. So, and then threonine. More science, yes. Right now we're trying to answer the question of basic cell and molecular biology. Draw the structure of any of the 20 amino acids and then of a dipeptide. And I'm just reviewing threonine structure. If you remember in the other video, this is the part I'm like, this set, I have to keep on trying. Threonine, CHCOO minus. Oh, I remember now. H3N plus. This is just the CCOH. C exclamation CH3. Nice. And then this one, non-polar. This is asparagine, and then this is glutamine. What does that look like? Asparagine looks like, hopefully we can write it properly. Asparagine looks like wait, C, C, and then you split. No. Yes. Ugh. I think this one is NH. Okay, no, I'm screwing it up. What does asparagine look like again? I'm close. Asparagine. C. Ah. Yeah. So it was a C. Who was the N? Later, we'll check. Asparagine is C, double bond O, and then NH2. There you go. That is asparagine. And then glutamine is just the longer form, so it's C, another C, and then you split and H2. Yes. Tyrosine. That's a phenylalanine. Okay, you have a CH2, and then this, and then OH. Well, cysteine is CH2 and then SH. That is where. So now we have 9 plus 6. What is it? 15. Now we just have to draw the polar charged molecules. Polar charged molecules were... We have... I know it's Ker. KRHDE. We have lysine. Oh my goodness. What was this one? CHCOO minus H3N plus. Lysine. Was this the weird one? Like CCNC? Yes, I think so. Lysine structure. Oh, I'm missing. No. It's 1, 2, 3, 4 C's before the N. 4 C's. C. 4 C's and then the N. C, N. That's a lot. So it's CH2, NH2. Meanwhile, arginine. So again, lysine is a CCCCN. Arginine, okay, this one has the thing mixed in. COO minus. I think this is CC. Okay, I should just make sure before I write the wrong one. Arginine structure. This one has switches it. Okay, where is it? Arginine is C. 3 C's, N, and then C. I see. So many C's. 3 C's, N, and then the C that splits. Okay. The arginine is the weirdest looking one. NH2, NH2. NH and then NH2. If I make it NH2, then that's a positive one. Alright, I have to erase them again. I just want to make sure I don't mix it up. So lysine is like this. H, C. Yeah, there's too many C's. So many. I will write C. Yes. H3N plus. Lysine 1, 2, 3, 4 N H2. Arginine HCCOO minus H3N plus 1, 2, 3 N, the C, and then you split into, oh yes, no problem. We're just going through it slowly but surely. And then NH. Trying to memorize the 20. Histidine. Another one that's weird. His and H. This one is HCCOO minus H3N plus Oh no. Well, no, we're fine. Ring. I think this is the ring. Is this one? CH2 and the 5 ring house. Something like this. And then N here and an N here. Oh, what does it look like? We're so close. Histidine. Okay. Yeah, we're almost correct. Bond. We're missing a bond somewhere. Histidine. I want to try and check the picture that I tried to draw before. We have a bond over... It's switching though. Where's the bond? This one's the bond. Histidine. I'm used to that bond being over there, but let's see. One of the pictures I'm looking at right now. It is the 5 house. Ring. The house. Bond. Oh no, that looks ugly. No ugly pentagons allowed. Here. Pentagon. Nice. So we have a bond. And an empty one. An N. And a bond. And NH. Ah, there it is. So bond, line, N, NH over there. Okay. Close. So now it wants... now we have aspartic acid. Aspartic acid and glutamic acid. This one, these are negatives. KRH are positive amino acids. So now, aspartic acid structure HC, COO minus, H3N plus. What do you look like? C? Another C? How many? 1 or 2? Okay. My main guess is just 1, and then it will split into an O and O minus. I think, but I will verify right now to make sure I'm not writing aspartic. I'm not writing the wrong ones. NH2 C's. Okay. CH2C. Nice. And this part can be OH. Oops, wrong. Can be OH. Meanwhile, glutamic acid is CHCOO minus, H3N plus, and a C, and a C that splits. That's why it's negatively charged. Booyah! Now, if we want to draw a dipeptide, let's see. There's so many C's. Okay. Dipeptide. Oh, so that's how it looks like if it's less than 2 minutes. I see. Yeah, that's the timer is funny. Timer. The red blinking one. It's okay. How can I... Let me just type it really quickly. No, it's fine. It'll be fine. Erase the timer. Nah, it can be erased. It can be. Draw the structure. Dipeptide structure. Dipeptide. Okay, so for dipeptide, how does it look like? Well, first let's draw an amino acid, H3N plus, C, COO minus, H. Let's just write glycine and alanine to make it simple. COO minus, CH3. Yeah, glycine and alanine. And then after this, I kind of want to draw this one properly. C double bond O, OH, yeah. How about U? NH2. You have an N. You have H. So what we're trying to do is get rid of this. Wait. We're trying to get rid of, where is it? This one. Yes. Yeah, there you go. And that will go out as what? 2 hydrogens and 1 oxygen is water. So that's why when you form a dipeptide, it's called what's that called? Oh, so now the ship is blasting off. I see. I wonder okay, if we increase it by let me just write it here, by the way. I'll fix that later. But yeah, in theory, the timer will normally increase. I think if someone subscribes and members and supas, but I haven't tested it. I don't know how to test it because it won't, like, I don't know if it works, so that's why I'm just testing it. And if I see that it's working, then nice. Okay, what's this called? Dehydration reaction. Dehydration reaction. That's why, because you're removing water. And the way the final output looks like, it looks like this. C the one O N H CH CH3 COO minus. See, this is a peptide bond. Peptide bond. Boom. That's how a peptide bond looks like. Now, it says draw the structure of any of the 4 standard DNA mononucleotides. Let's see. DNA mononucleotides. Oh wait, water. Okay. DNA mononucleotides. Wait. 4 standard DNA mononucleotides. Right. Oh wait, actually, this is a good stopping point so that I can save the DNA stuff later. As for now, I think I need to rest my throat. Okay. Oh no, actually no. I think we can just continue it. I can do it. Whoa. DNA mononucleotides. DNA mononucleotides. And indicate the position that distinguishes DNA from RNA. Okay. So if we write this one, oxygen, DNA is deoxyribonucleic acid, right? The base. Then you have an OH here, CO, the phosphate, P, P, and an O minus, double bond O minus, double bond O minus, double bond O, and then O minus. We don't have space, so that's why I'm making it curvy. Meanwhile, this is RNA, mononucleotide. RNA would have OH over here as an extra, because we talked about it earlier how it is more reactive compared to DNA. So deoxymonophosphate has a phosphate group at the 5' carbon of deoxyribose. And then DNA lacks a hydroxyl group at the 2' position. In a dinucleotide phosphate, the phosphodiester bond links the 3'. Where is it? It links the 3' OH of one sugar to the 5'. Where is it? phosphate of the next one. So imagine if you have a next one here. Here, OH, O and phosphate, O minus, O and squibbly. That will connect with this one, the 5' phosphate of the next molecule. So that is how it is connected. And then the next question would be, why are DNA sequences usually shown in the 5' to 3' orientation? Well, wait, water, earth, fire, air. There, 1 second. Okay. The way water works. No. The way the 5' to 3' direction works is like this. Let's erase it first. 5' to 3', and here. No, not the red one. On this one. 5' to 3'. DNA sequences are usually shown in that orientation because DNA and RNA polymerases synthesize the nucleic acids by adding the nucleotides to the free 3' hydroxyl group. Where? It is here. It adds it over here. This is the star of the show. 3' hydroxyl group. So you add the new ones there, and then add the new ones to the 3' hydroxyl group of that one. Add it to the 3' hydroxyl group of that one. And that makes it the natural direction. We talked about how introns were originally considered junk DNA. And the reason for that is, well, introns, because here's the thing. Exons and introns. Exons are the ones you keep when you splice, but introns are usually you get rid of it. But now we know that introns allow alternative splicing, and they contain regulatory like elements which influence the gene expression timing. Okay, but I think so far I really need to rest my muscle. I have been talking a lot and I've been yapping. But yes, thank you very much for coming. I will rest first a little bit. But yes, that is biology so far, and more to come hopefully. Thank you for watching. Bye bye.